Okay, welcome to our zoological excursion in the master program. The zoological excursion is done on 2 days. Of course, with this mode this year and the corona outbreak, we do it virtually, and yeah, we do it not on consecutive days, but we will meet for several sessions. And you can follow everything virtually. We will present all the material, and you can then look at it at home. This is unfortunate, of course, because you will miss all the atmosphere. And this brings me right away to the place where we are. Well, we have 2 days for zoological excursions to introduce you into the diversity of life, of animal life, in the surroundings of Goettingen. And of course, this is totally not enough, because we have to select some places where to go. And actually, we selected places which are hotspots of diversity, not just in the vicinity of Goettingen, but basically all around Europe. This is an anthropogenic habitat, so we're intentionally not going into forests which are close to the natural vegetation. Of course, forests are also very diverse, but from a especially conservation perspective, it's not the forests which may need major attention, but it's the open habitats. The open habitats are formed by man, as just mentioned, and of course there is habitats from a conservation perspective which are kind of very valuable, and others which are of very low value. So we're of course not in an arable field. We're on a meadow, or a former pasture actually, and this has been formed by pasturing, so by grazing animals. And this brings me a bit into the history of basically grazing and pasturing in Europe. Actually, this is probably the oldest way of land use in Europe. It originated in the very south, Mediterranean region, and it was mainly based on sheep, at least initially. And this sheep grazing then spread from the Mediterranean region basically all across Europe, even to northern countries. A bit more about the history. The grazing history of Europe is based on basically shepherds taking their sheep through the land. And the typical thing is that these shepherds, they were moving across the land. So they were moving with their herds, typically from regions where they stayed during winter, a warmer place, to places which were less dry in winter, so typically higher mountain regions, where the pastures were present in a good way for grazing, so with green grasses, also in summer. This is a typical nomadic lifestyle which today disappeared basically in Europe, but it's still present in various regions of the world. So what we look a bit into is historically built landscapes. And this applies to these open, dry or grasslands, which until today are not heavily used. Many of them today are conservation areas, so protected by law. This is actually not the case where the place we are at the moment. Somewhat west of Goettingen. It's a hilly area, as typical in the vicinity of Goettingen. It's actually a limestone area. This is Triassic limestone, which is also a typical deposit all around Goettingen. And it's a south or southwest facing slope. so it's a rather warm place. And this, of course, is also the reason why we find a lot of species which basically would be either not present or would have a very much more restricted range size in size of areas where they occur in Germany and in Europe, if there wouldn't have been the pasturing of the ancient times. And actually this pasturing, this continued until today. I mean, there still is sheep pasturing, shepherds in the also surroundings of Goettingen. Not many. Most of them are kind of employed for conservation reasons. The area here is more, today is more used by horses. So another way of pasturing, and it's also of course different to the previous times, because the pasturing today is based on stapling, basically, so fencing the sheep or horses to one place. So this is quite different to what it has been. Well, a few words to the difference between pasturing and meadowing. So what we find much more widespread today is hay meadows. This is also currently kind of ongoing in the vicinity here. So we have the first cut because, well, today is a late day in June and the first cut of the meadows just started. But from a conservation perspective, grazing and mowing may cause quite strong differences. Mowing in the end results in a more homogeneous sward, and grazing causes a lot of microhabitats which are used for from quite a number of different species. So the heterogeneity of the habitat is a very important aspect of the basically diversity, and this is why also for in a number of protected areas, it's still that they are grazed by sheep, and the shepherds are paid basically for doing that, because from an economic perspective, this of course doesn't pay anymore. A little bit more into this difference, the change in the use of the land in the last few centuries, because some 200-300 years ago, it was really that most of the land in the vicinity of Goettingen, and actually also applies to the more hilly regions in Germany, was used for pasturing, and it still were nomadic people who were moving across the land. This was, by the end of the 18th century, resulted really in a degradation of the landscape in Central Europe, and the forest was down to below 10%, and most of the region was really open pasture land. It is actually assumed by many biodiversity-oriented people that it was this time when biodiversity in Central Europe was at a maximum. And since these times, probably diversity declined. And this applies in particular to especially these open habitats which had been grazed, and the grazing then disappeared. It was turned, of course, into either arable land, or it was turned into hay meadowing. But all that presumably resulted in a decline in diversity. So what we look into is a bit the history, how much of the diversity in Central Europe kind of came about, and we want to get a bit of a view into all this diversity. So this is the goal, and of course if we talk about diversity, we in animal diversity we talk mainly about invertebrates, and in the invertebrates you all know this is mainly insects and a few other arthropods. It's a great diversity, and it's quite tricky to get really a hand on. And what we try to do with these excursions is to get you really a field feeling for all these organisms. Of course, you can also do that, and you may have done that in your bachelor studies, that you determined species in the lab. But if you're out in the field, you often cannot do it exactly the way you do it if you would have a dissecting microscope. You just need for the field a lens. Typically such a lens is fine, 8 to 10 times magnification, but this is actually essential. Otherwise you will miss and be unable to see the really many, if not most, of the important characters. So you really need a lens if you ever want to do some really field faunistic work on all these arthropods and other invertebrates. And what we also try to do is to provide you with more field-based characters. This is often different to what you have in a typical dichotomous key, where you go through one by one. In the field, it's often actually much easier than that. If you know something about the fauna, you may need just a few characters, and then you may be even able to determine species really to the species level. Of course, there is a lot of restrictions here, and we will also mention that, but we will do our best to introduce you into these kind of field characters. We are actually here not alone, but there's 3 more people. It's Mark Maraun, it's Johannes Lux standing behind the camera here at the moment, but he will help very much, as you will see, with the determination, but also with all the documenting of all the animals. And it's also Svenja Meyer, also helping a lot in determination, sampling, whatever. And yeah, we'll come back to those later. So in addition to the grassy area, the meadow, where we sampled, we actually sampled also in some specific habitats, like this one here, a rocky place, so also an open habitat with very little vegetation, in part basically none. Here, actually at this place where we are, this is purely anthropogenic. This is a limestone excavation, so the limestone was used for construction or whatever. So this is a purely anthropogenic habitat. It's very interesting, and it resembles also some natural habitat, which is kind of rare in Central Europe. So only along, for example, rivers, where you have steep slopes and some of these limestone kind of escarpments. Well, in close vicinity here, for example, a bit further up in the Werra Valley, you may find such kind of limestone escarpments. Close to Eschwege or Treffurt or Kreuzburg. This is very nice places, very interesting places, and I would highly recommend going to these places because you will find some species of insects and spiders which kind of are very characteristic and only occurring in such sites. Well, they often are more widespread further south, so this is also a bit an untypical place where they occur, and it's an anthropogenic place, very similar to what we saw with the meadow area, which is of course also an anthropogenic habitat. So such habitats are of course more widespread and characteristic of Mediterranean regions or also the alpine regions. Yeah, it's interesting places because of course there is very characteristic environmental factors here, also tricky things from a nutrition perspective. There is no grasses, very little primary production, so the species dwelling here rely on other resources and have to cope with very harsh environmental conditions, very high temperature conditions, low moisture conditions, and so on. So it's from that perspective also a very interesting habitat. In our zoological excursions, we sample all the animals, I mean the invertebrates and insects, by very simple means. You may also simply pick some by hand or by using some scissors and place them into these schnappies. But it helps a lot if you do use some devices like this, which allows you to collect whatever is in the vegetation. So we focus, of course, of all the animals which are above the ground and able to crawl or fly in the vegetation. So we take these samples all across the sward here. You may also sample close to the edges of the sward of the meadow, where there is more trees and bushes, and also look a bit into the ground, into the soil and litter layer. You may also look a bit into dead wood, turn around dead wood if it's there. And in addition, there is a specific habitat here. This is stony habitats, really open, stony places. This is also anthropogenic, actually. So it was a excavation of limestone, of this Triassic limestone, for building houses or whatever. Now, I'd like to guide you a little bit to what we have found, and we will present what we have found in a phylogenetic context. So we'll basically follow phylogenetic relationships of the different groups which we found. And we will start off actually with presenting a few species we found in this very large group of the Lophotrochozoans. So Lophotrochozoa, we found a few mollusk species, gastropods, and a few annelid species. It's one lambricid species. So this is one unique, very large group with mostly marine groups in it. Then we will move on into another very large group of the animal kingdom, and this is the ecdysozoans. And of course, in the ecdysozoans, we will basically only present the arthropods. Radiation of arthropods is absolutely amazing. Starts off in the marine system, but we will see there is at least 4 transitions to land in the arthropods, and of course we will deal with these terrestrial living groups. And we will start off with the basal groups, basal group in the arthropods, in the euarthropods, or true arthropods, which are the Chelicerates. And there we have the transition to land in the arachnids, and we only will present one group of the arachnids, and this is the spiders, the Araneae. The sister group of the Chelicerates actually are the mandibulates. And in the mandibulates, we learned quite some new structure in the last 10-15 years, and we're now confident that actually at the base of the mandibulates are the myriapods. So this is an independent transition to land, and today all myriapods live on land. Well, we know very little about this transition, but we're rather confident that they made the transition to land independently from the other groups in the Chelicerates and also in the crustaceans. Yeah, this brings me to also a kind of a new grouping with the sister group of the myriapods being the pancrustaceans. And this is kind of a renaming. There's still the crustaceans in there. I'm sure you all kind of know that we now assume insects basically to be a group within the crustaceans, and this of course results in crustaceans being paraphyletic, and the new kind of monophyletic unit now is the pancrustaceans, including the former crustacean group and the insects. Again, we have two transitions to land, major transitions to land. We have one major transition to land in the crustaceans in the isopods. There's a number of other transitions, but it's most pronounced, of course, within actually this group of isopods, where we also have a lot of marine and also freshwater species, but we have also a lot of terrestrial species. And then, of course, everyone knows there is the insects, the most diverse group of all, for sure. And the independent transition of transition to land was made by probably small crawling six-legged species of what later became insects. We start with presenting our collection of today in a phylogenetic, of course, sequence. So you can see the different groups we encountered here, and we start off with a group with only few species we sampled, but it's of course a tremendously important group with mostly marine taxa, and this is the Lophotrochozoans. The Lophotrochozoan. We have two terrestrial groups here, the mollusks and the annelids. And as said, we only have few species. Well, there certainly are more mollusks here, but we only found actually two. I mean, it's a dry grassland, so there aren't too many mollusks to be expected, because basically both mollusks as well as the annelids, they depend on water or they desiccate easily, so you wouldn't expect too many of them in this open dry grassland. And in fact, the two mollusk species we found, we collected at the borderline to the bushes and forest. Yeah, both are actually very abundant. The one is a Clausiliid mollusk. This is a family of mollusks which has a very elongated shell, and the opening has teeth. So it's very characteristic actually, and this one is one of the most abundant species of the Clausiliid mollusks, gastropods. It is Balea biplicata. You can recognize this species by the longitudinal kind of furrows in the shell, and also the tip of the opening is a bit narrowed, tip-like. Yeah, these Clausiliid gastropods you will find in basically all our calcareous woodlands, in part quite abundant on stumps, or also often under logs. So a very characteristic group. And the same actually applies to the next one. This is Discus rotundatus, a small flat shell, and this is actually of the family Patulidae. It's very characteristic also, especially underneath of logs, close to stumps of trees. Yeah, mostly active during wet periods. So again, somewhat untypical for the open grassland, but at the edges you may find it. If you're actually interested in mollusks, there is a nice, rather recent new book, the Landschnecken Deutschlands, from Vollrath Wiese. This is a really nice book, very helpful, covering virtually the full spectrum of the Central European fauna. So this is a very useful book. that's it for the mollusks. A group you probably also wouldn't have expected in these dry grasslands are earthworms. But in fact, they're everywhere. And they're quite abundant actually on these, even on these dry grasslands. They're only active during the winter month, and in summer they hibernate. And this actually also applies to Mediterranean regions. So even in warm, kind of dry places during summer, you may well find not really species rich, but quite a few species of earthworms. Well, the Central European fauna is kind of species poor. We have some 40 species of earthworms in Germany, and they might be divided into 3 ecological groups. We only found one species from one of the ecological groups, and this is the so-called endogeic groups. The endogeic group. Endogeic meaning they live in the uppermost mineral soil. And this actually is one of the most characteristic species in this endogeic earthworms. It's Octolasion lacteum. One of the most abundant and characteristic species in this endogeic earthworm group. And this is actually a juvenile, and juveniles of earthworms are hard to determine because the key relies on the location of the clitellum, and of course as a juvenile it doesn't have a clitellum, so no progress with any key. But in fact these earthworms do have, despite their paucity in characters, they do have coloration, which is very helpful for determining them. And all these endogeic species actually are poorly colored, so they are not very reddish, but rather pale. And this applies in particular to this Octolasium, and well, lacteum meaning milky, so it's whitish. And also another very characteristic feature is Octolasium, the genus Octolasium, which is with 2 species in Central Europe, is having mucus glands, yellowish, kind of yellowish segments by the end, and also in the first kind of third of the body. An interesting aspect of this Octolasium is that it's purely parthenogenetic, so it doesn't reproduce by sex, and this is fairly common in earthworms, despite their hermaphrodites. So the typical reproduction is as a hermaphrodite. Quite a few species do not mate and just do it without mating. Okay, this brings us to the main diversity where we collected species from, and these are the arthropods. Arthropods, as you I'm sure all know, form part of the ecdysozoans. So one really big terrestrial phylogenetic lineage, which is monophyletic. And in the arthropods, the basal lineage are the Chelicerates. The Chelicerates, we mainly found spiders, and these will be presented with some introduction by Johannes Lux in a minute.
So, hi, my name is Johannes, as Stefan already told you. And also, as Stefan already introduced, we now move on to the Chelicerates, forming the sister group of the mandibulates and the most basal group of the arthropods. And within the Chelicerates, there are two major groups of recent Chelicerates, which are the Xiphosurans and also the arachnids. But in terrestrial habitats we only find arachnids because Xiphosaura are also called the horseshoe crab, and they're entirely marine organisms. So this is also something we then can suggest that there was a transition to land by the other arachnids. And within the arachnids there are several groups, but we only found one group, which is also the most diverse group of the arachnids, the Araneae, the Webspinnen, weaving spiders, or spiders basically. And yeah, within the spiders, there's also again split up into major groups. There are the orthognates and the lepidognates. The name, as the name suggests, it's a bit like the orientation of the Chelicerates, which is leading to that. So it's major for the orthognates. They move the Chelicerates just oriented with the body orientation, so in front. And lepidognates can use their chelicers against each other. So yeah, I may mention that for sure the Chelicerates have those chelicers, which are mainly scissor-like structures which are used as mouth parts. And for the Araneae, for the Webspinnen, they are a bit reduced, so there's only still one segment which is movable, more or less. So they look a bit more like a knife and not like a traditional scissor. Yeah, and we only found lepidognates here. And within those lepidognates, there is an quite old taxonomy, which is not very correct at the moment, but I may move on with it because we found a cribellate spider. So this is the structuring in cribellate and ecribellate spiders, which is referring to the cribellum. The cribellum is a small plate in front of the spinnerets of spiders. So this small plate also have a lot of silk glands, and spiders can weave also silk with this cribellum. And with this they do not need glue points anymore. So if you think about a traditional spider, you may also think about very sticky silk, but those cribellate spiders, they do not need the silk because they weave such a fine and fine structured silk that the prey is just entangling in within without use of glue points. This is also something which also a bit refers to the habitat. You can mainly find cribellate spiders in drier habitats because the ecribellate spiders more or less need water to produce those glue points. And here it's quite a family which fits to the habitat we saw before. This is the family Titanoideidae. The Titanoideidae are, maybe we could call them like limestone spiders, the Kalksteinspinnen. And so they are easy to determine because they are small, black, but the prosoma, so the former body, the first body part is red, so red colorated. And the opistosoma is blackish for the females. And in this species, Titanoidea quadrigutata, we can see that the male possesses 4 white dots on the opistosoma. So it's quite easy to determine when having a male. When you only have a female, it's a bit more difficult. Yes, but very interesting spider, just sitting here in those stony habitats, building more or less a sheet web, but which is vertically oriented within stone or between stones, so in the free space between stones. And they have a funnel downside, so they just wait for animals just crawling over the stones, falling when they get entangled in this sheet web, and then they directly fall into the spider's home, and then they can be they are killed. It's also another thing I may mention. So all the Araneae, all the Webspinnen are poisonous. They use poisonous glands within the chelicers. There's one family, the Uluburidae, which do not possess them, but they are reduced. So it's really an autapomorphy. Yes, and then we move on with ecribellate spiders. And this is basically one family you all may have encountered. It's the Araneae. It's really the typical family you think of when you think of a spider. They're building a wheel net. So you may all have seen them. There are also some other families building wheel nets, but one family you may mistake them with by net are the Cheiracanthiidae, but they weave an open, they leave an opening within their net, and the Araneae do not. So the base of the weir net is closed. Yes, and when you see an aranid, you may also see some characters. The Araniidae, they really are quite spiny, so all legs are quite spiny. The third pair of legs is quite small compared to all others, and also they have a very iconical eye positioning. So this is also one thing you may encounter in the key. You always are asked when you determine spiders to look into their eyes. And yeah, here it's more like quite a square positioning of the eye, of the middle eyes. Yeah, these spiders have 8 eyes, and yeah, you may see it on a picture we show you. And yeah, what is interesting about this species, so this is Mangora acalypha. It's easy to determine because they have 3 rows of black dots on their opistosoma, which are connected by very thin lines, and it's really easy to see them. And the color is more yellowish. And yeah, they also have some interesting ecological facts about them, because they were also found to do ballooning very often. So what is ballooning? Ballooning means that a spider, especially spiderling, but there are also some adults doing it, spin a silk line which is captured by the wind, and then they continue spinning it, and they continue spinning it as long as there is enough power to pull them away. So they just fly with the wind, and they can even fly very high, and forming this more like air plankton. And there you can find often this species, Mangora acalypha. Yeah. And yeah, now we move on to another quite derived family. So these are the Theridiids, die Kugelspinnen. As the German name suggested, you can identify them, or a lot of species you can identify by their spherical opistosoma. The opistosoma, very spherical. The spinnerets are often orientated below the body. So if you look at the spider from the side, you may see the spinnerets more under the body. But there are also some other species and other families having that, but it's a good hint if you see that. Additionally, a lot of the species of this family are quite unique in their coloration, and you can determine them quite good. So, and here we have a spider where the general coloration varies a lot, but there are some parts which are really equally correlated between specimen, and this is Enoplogonata ovata. And what you can see really easy is that they have a very thin line on this spherical opistosoma, and it's just in the middle of some black dots. So there are often black dots, and you see this thin line, and it's often often also encountered by other horizontal lines. And there's also an interesting thing about the web I forgot. So they built a sheet web. All Theridiids do it. They have it in common with the Linepheids, also another very derived group of spiders. And they weave a sheet web, and then they have their lines vertically oriented. They can be oriented to the upside or to the downside. Depends a bit on where this species live. Here, for example, we have a species you commonly found in bushes or higher grasses or something. So they normally orientate them upside, to the upside, because they're actually aiming for flying insects to entangle. So the flying insects come, entangle in those silk lines, and then just fall onto the web, sheet web, where the spider is sitting beneath and then taking up the prey. Yeah, so very interesting species. And we also have another species of the family. This is Philoneta, and this may be probably Philoneta impressa. And there's also another species, but both species show very unique coloration. So we have a brown dot in the middle of the opistosoma and two black lines on the side, but they are also interrupted by 4 white lines. And when you see this combination, and it's also quite a big pteridiid. Most pteridiids are quite small, but these ones and also Endoplugonator ovata are quite big, like around 6 millimeters in length. And yeah, then you can identify the genus Phylloneta, and yeah, this may be Phylloneta impressa. And what's very interesting about this species is the child care. So this is something which is very prominent in a lot of spiders, but it's very, very interesting in the pteridiids, because some pteridiids, as this one, they live in the same web with their spiderlings. So they breed the spiderlings within their web, and then they live there with the spiderlings and catching prey, and even starting feeding their spiderlings. So they're feeding their young. So they're sucking up prey, and then they just puke it again to the spiderlings, and they suck it up again. And this mother care, female care, even goes further, because at the end, when the spiderlings are about to leave the web, the female just set a bite on herself and kills herself and will be sucked up by the spiderlings. So interesting story, but from a human perspective, quite horrifying. And I told you about a bit about the web structure of the pteridiids, and here we have individuals of the family of the species Episinus angulatus. And this is quite interesting because they reduced their web very much. So this sheet web with those lines was the basal web in this family, and they derived it very, very much. So they only used 2 silk lines connected with each other with another silk line, and just waiting actively for prey. So they just connect those silk lines to their forelegs, sitting beneath low-structured grasses or stones, and waiting that another arthropod crawls by, and then they just wrap it, or they just pull them up and wrap them and store them for eating. So it's really, really interesting. And you can easily identify this species because the opistosoma is not spherical as in a lot of other pteridiids. It's more triangular in shape. And if you see an pteridiid with a triangular opistosoma, it is mainly this genus Episinus. So now we continue with some free hunting spiders. So we had spiders building webs, and now there are also a lot of free hunting spiders. They can still spin silk, but here's a family which barely do it. So these are the thomisids, the crab spiders, Krabbenspinnen, and as the name suggests, you can just identify them by looking at their general habitus, because the legs are oriented to the sides and they mainly walk as a crab would walk. And yes, and the hind legs, so the 2 pair of hind legs are really small compared to the forelegs, which is really determine this family. And there are numerous species and numerous genuses, but here we have some small brownish ones. I also will show you another species in 1 second. And those small brownish species, they are mainly belonging to the genuses Ocyptilla and Xysticus. And what you can do to really get a hand on those genuses and to know how to determine them, or to not mix them, is when you look again at the eyes. So Ocyptilla, their middle eye square is higher than white. This is really a good trait, and you can also see it, but you again may need lens. Yes, thank you. Yes, and the specimen of the genus Ocyptilla. So we have here an Ocyptilla, and the species within the genus are very hard to determine. So we stay with Ocyptilla, and they live more on the ground, which is also unusual for crab spiders, which mainly can be found in vegetation and also on flowers waiting for prey, because they are sit and wait predators, waiting for another arthropod crossing by, and then they catch it. And yeah, the genus Ocyptilla mainly sit on open fields and wait for example, ants come by and then they catch them. And what's also good to see is that there are often club-like setae on the body of Ocyptilla. So if you see a small brown arachnid, small brown tomocid with those club-like setae, you can for sure say it's Ocyptilla. And now we come to this other genus you may mix Ocyptilla with. So this is Xysticus. This is by far the most common tomocid. And yeah, again, legs oriented to the sides, crab-like, but here we have the case that the eyes form a real square, so they are as long as wide, or they are wider than high. So you also again have to look at this middle eye square of those 8 eyes. And what's interesting and what we see, because Xysticus is more sitting on branches, on dry vegetation, and they mainly catch prey which is by far bigger than them or their size, because they seem to have a very well working poison, especially for arthropods. And what they do is they sit somewhere, also on flowers, or some species on flowers, and then they wait until pollinator encounters the flower and they just bite. And they always directly bite behind the head. So they bite in what we would call neck, and they kill basically this pollinator immediately. And here we have also the case that we see this spider here killed a bee, and and it still feeds on the bee even though we enclosed it. So yeah, rigorous predators. Now we come to a family you may mistake with the tomocids, because this family also have legs which are oriented towards the side. Also have a bit like crab-like appearance, but they really do not move to the side. They always run directly frontal. And these are the philodromids. And they also, you can also determine them when looking at those legs. So legs are oriented to the sides, but they are not, so the hinder leg parts are not as reduced as within the tomocids. Basically, the legs are more or less all equal long, not truly, but it can appear like that because the second pair of legs is the longest, but yeah, they are quite equally long. And yeah, free hunting spiders, and also a very difficult family, because this family is also a difficult genus and family, and because here we have the genus Philodromus, and this is also again a very common genus. All spiders are there quite brownish, not really having any good traits to determine the species, except for the epigyne. This is a case in almost all spiders. When you want to determine the species, you may have a look at the sexual traits, which are for the males the secondary sexual organs, which are called pedipalps, and for the female the primary sexual organ, the epigyne, which is the covering of this primary sexual organ, which is called the epigyne. This is a sclerotized plate sitting on top of the opening, and it really works like with a key principle. So the males just fit in the females of their species, and this helps often by determining spiders. And what you may also see in philodromids, they have 2 rows of small eyes. And I didn't told you anything about genesis traits here because it's quite easy, because all other genesis of this family are quite rare around here, and they are also colored different. So when you see a philodromid in brown, it's for sure Philodromus without any interesting markings on it. Yeah, it's Philodromus. So now we move on to another species and also another family, the Gnathosidae, which are called Glattbauchspinnen or Plattbauchspinnen. And in English they are called ground spiders. And how can you determine this family? Or how can you identify this family? It's quite easy because you have to look at the spinnerets of those species, and you see the spinnerets are quite cylindrical. So they have very, very long spinnerets and cylindrical in shape, and they basically have 6 spinnerets, but 2 of them are really reduced and in the middle of them. So it looks like there are 4 large cylindrical spinnerets just diverging at the end of the abdomen. And most of those species of this family are quite colorless, but there's also a group which is only blackish, and this is this group around Zelotes. There are also some other genesis as this one, Tachycelotes, and they are quite difficult to determine. But with this species we had quite a lucky shot, because Tachycelotes pedestris is easily to identify by its legs, because the legs, the tarsal segments and also the metatarsus of the legs are reddish, and all other segments and the whole spider is black. The rest is black. And what's very interesting about these spiders is that the Gnathosids are thought to be mainly nocturnal, but this is true for all species. A lot of them are day active, and they hunt for mainly ants. So there are a lot of species which are quite specialized on ants, and they can directly bite into the bases of the antennae of the ants, and they really just approaching the ant, setting this gift bite, and then this poisonous bite, this poisonous bite, and then they just run away and wait for the animal to die and take it up again. And very easy to identify this species. If you see a Gnathosid with this blackish body and reddish legs, it's for sure Tachycelotes pedestris. Now we move on to very, very large and also quite horrifying looking species of same family, so also Gnathosid, but this Gnathosid is more brownish in color, and it belongs a bit to a genus group, because this genus Drasodes was divided several times in the last 5 or 6 years, and so you cannot easily say if this specimen here is from the genus Drasodes or Drasodex. But what they all have in common, that they are very large, they are brownish in color, and they are really typical for those stony habitats. They sit under stones, so they are really nocturnal, and they weave a silk sac to rest at the day, and then they come out at night. And what you may see when you catch such a specimen, they are quite aggressive, and they are also thought to be mainly hunting for other spiders, because when you see them at night you also find them underneath stones, but just because they're actively crawling under those stones, and they are thought to just crawling into the nets of other spiders and kill them and eat them. And so if you see such a large Gnathosid, which is around 15 or 16 millimeters in length, and it's a Gnathosid, you may say that it's Drasodes or Drasodex. Yes, and they also have some kind of a female care. You can also often, especially when you, in the summer month, when you encounter the species, you find this silk sac, and within the silk sac you may also find an egg sac, which is really which is protected by the female. The female is just sitting with this egg sac until the point the spiderlings hatch, and then they leave their mother and just distribute in the habitat. So, and now we have a very interesting family, and also a family which is very well known also to the public, because it's the Cheiracanthiidae, die Dornfingerspinnen. And most of you may know one species of it. It's Cheiracanthium punctorium, the Ammendornfinger, which is one of the few species in Germany which is thought to actually can hurt humans, so can just pierce through the skin of a human. But they rarely do, and it's not really good story. But here we found a juvenile of Cheiracanthium, and you can easily determine them because they also free hunting, building egg sacs. They look a bit like clubionids. Clubionids, they have conical spinnerets. And the prosoma often looks like it's cutted in the front, so it looks very stout at the front. But you can not mix it up with the clubionids, because if you you should take a look at the forelegs. When the forelegs are the longest pair of legs, you can for sure say that it's Cheiracanthiidae. And within the Cheiracanthiidae, there's also there's only one genus you can encounter here, and this is Cheiracanthium. But because this is juvenile, we cannot identify it further. But what you often see in the genus Cheiracanthium is also that they have a cross-like marking on the opistosoma, so you can really not really mistake it. Yes, and they're also free hunting, as I said, building egg sacs, and they just walk quite slow. Those forelegs are pointing to the front because they use them a bit homologous to antennae. So when they encounter prey with their forelegs, or they touch prey with their forelegs, they just rapidly attack the prey and kill it. Yes. Now we come to quite a highlight family of these spiders, because some of you may know them, and they are quite cute. These are the Salticid spiders, Salticidae, Springspinnen, or jumping spiders. And you can easily determine this family by looking at the eyes. The two front eyes, or the two mid eyes of the front eye row, are really huge. You cannot mistake it. They are really large and give this animal quite a cute appearance. And as the name says, jumping spiders, those spiders are able to jump. You often may see them also on the wall of houses, and then you can see how they getting their prey. So they're just approaching, so they're really visually orientated, so they take a look, and if they see prey, they approaching near to it, they some kind of sneak towards it, and when they're in the right position, they directly jump on their prey. And those jumps may also miss. This happens quite often, that they do not catch their prey, but just jump anywhere. And for this case, they always spin one line of silk, which they attach to the surface, which work a bit like a bungee, Seil. And so they're really safe when they're jumping. And here is a genus of this family, which you can easily determine when you have a female. It's called Heliophanus. And as the name said, Helio, which is referring to the sun, because their pulps are yellowish as the sun. And the females have yellow-colored pulps. This is also quite interesting for the Salticids, because normally it's like the male are colorated a bit more colorful, a bit more shiny, because they are really sending optical cues to the female, and also some species specific optical cues. But in this case, the female is a bit more fancy colorated with those yellow pulps. I don't know really why, but yeah, you can easily identify the family and with the genus. And within the genus, it's not so easy. You again have to look at sexual traits, but the problem is that we have a juvenile again, so we cannot do it. So we stay with Heliophanes. And now this is quite a highlight species in the end, which is really mainly Mediterranean, but you find it also on those stony habitats here. This is Elorillus V. insignitus, so or V. insignitus, and this is a salticid, and this name is referring to a marking of the male, because again here is the case that the male is quite colorful, and they have a V-like shaped marking directly in the front of their prosoma, directly above the eyes. So it looks like a V, so they look quite angry all the time. We may also show you a picture here. And so the male is really easy to identify. And here we also have a female, and this female is a bit more colorless, so it's brownish general in color. But what's the really important character here is when you look at the side eyes of the first row, directly beneath it there is a white stripe, and above this white stripe there's often some kind of reddish or brown reddish coloration. And when you see those markings on a jumping spider, you can for sure say it's Elorillus V. insignitus or V. insignitus. And again, here they really need open habitats. It's again a thing, a lot of salticids need those open habitats. You barely find them in forests because they're optical hunters and they just hunt by visual clues. Okay, so as you see, I just changed my clothes to present you another book because there is a determination guide for spiders in the field, which is quite good. This is the Spinnenführer from Heiko Bellmann, and this is a very nice book, nicely pictured, also some drawings within to differentiate between species, and also there is some kind of a field guide for the families. So it's a really good book, and something you should have when you want to determine spiders in the field.